Students, today's practical session is titled Root Anatomy of a Monocot Plant. Now, this is a monocot plant. Grasses are example of monocot. This is a grass, and the name of this plant is Setaria valvata. Now, we want to see the internal structure of the monocot plant. So, the materials we will be using, we have our monocot plant. We have a series of ethanol from 50 to 100%. We have a saffronin stain, we have a glycerol, we have a nail varnish, we have a slide and cover stick, then we have distilled water in petri dish, sodium hypochlorite in petri dish, and distilled water in petri dish. So the experiment paper. Now we are going to be doing freehand sectioning of a dicot of a monocot root. So you can cut small portion of. Now we're going to be using a styrofoam material so that we can have a kind of tissue block, so it will hold this material tightly, forming a kind of tissue is so blocked. So we are trying to using this method to mimic a microtome machine, which is used in sectioning tissues and cells. Now we make several cutting, transverse section, several cutting from right to left, several cutting into distilled water, and then make sure that what we are cutting is we make a very thin section, so that when we check out the microscope, light will be able to pass through a thin section. But if it's thick section, light will not pass through. You won't be able to see your organelles very well. Now, this is a thin section. You just cut several cutting. As you make several cutting, you transfer it into the water. You make several cutting, transfer into the water. They are in the water, though they are very, very small. You can cut into distilled water. Now we cut in the distilled water so that we can hydrate our cells, so the cell will not become flaccid before we start staining and giving us a poor result. So after the sectioning, you put a drop on a glass slide. You can drain some of the water. You put a drop on the glass slide, and after putting distilled water, you now put a drop of sodium hypochlorite to decolorize the section. Then you can remove a little bit. So this sodium hypochlorite can stay for like 5 to 10 seconds to decolorize to decolorize. Then we now transfer back into distilled water. You put a drop of distilled water to remove the effect of the sodium hypochlorite, so that when staining, it will not interfere with our staining. So we are ready for the hydration now. So can I put a drop of 50% ethanol and leave for about 5 seconds. So we put a drop of 70% ethanol. We are dehydrating gradually from 50 to 100%, so that our cells will not break, so we can have proper staining. Then we also put a drop of 100% ethanol. Then you can drain the excess. Then it is ready for staining. So you put a drop of saffron in and leave to stay for 30 seconds. Make sure the stain covers the cell, and leave to stay for 30 seconds. After 30 seconds of adding saffronin stain, you put a drop of glycerol for easy visibility under the microscope. Then we cover still from one side. You can put even 2 drops of glycerol, 1 to 2 drops. Then you cover your prepare sample, your cut section with cover slip gently from one side to prevent trapping of air bubbles, and you gently lower it. You can see that there is no air bubbles formation. So you drain the excess stain. You make sure that your sample that you prepared is contained inside your stain. Sometimes, because it's a very fragile sample, you may want to move away from the stain. So you bring your sample to the middle. Yes, your sample that you prepare must always stay at the middle of your preparation. This is a very small section because it's the root, so you have to be careful. Your sample can move away from the stain. So after this, we add glycerol for easy visibility under the microscope. After the stain has been absorbed by the organelles of the cut section of the root, we add our glycerol for easy visibility. Then the final one, we add our nail polish. We seal the edge, or we rinse the edge with nail polish so that our sample will not be dehydrated. It won't dry before we start examining under the microscope. So you clean the base and examine under the microscope. Other times 4 objective scanning objective and times 10 objective for bigger view, and also you can check other times 40 to see more internal structure. Thank you for watching. If you find this video useful, please kindly like and share and subscribe. God bless you.